Okay, so this is the part that I promised you on equilibrium and head movement. So equilibrium, to me, this is it's a pretty interesting topic because it's you don't think about it. What is actually how you actually figure out like where you are in space and how you're moving. Okay, so it's based on the idea that you have to be aware and be constantly monitoring where your head is, like how your head, if it's forward or if it's backwards or if it's sideways. Okay, so the part of your ear that monitors this is called the vestibular apparatus. And it's the utricle, the saccule, and the semicircular ducts. Okay, so this information is going to be sent to our brain. Okay, but at the same time, our vision and the proprioceptors in your joints are also sending information in. Okay, so it's going to help us figure out, okay, so the way it's broken down is the utricle and the saccule detect static equilibrium and linear acceleration, which we'll talk about, and the semicircular ducts detect angular acceleration. Okay, so static equilibrium is when the head is stationary. So the head isn't moving. And then linear acceleration is if you're tilting your head. And then angular acceleration is if it's spinning. Okay, or yes or no, or like if you're a figure skater and you're spinning. Okay, so static equilibrium and linear acceleration. Okay, so the thing that detects this is called the macula. Okay, so it's the receptor and it's found in the utricle and the saccule. Okay, so here's the inner ear. Okay, here's the cochlea. And then here are the semicircular canals. And then at the base of the semicircular canals, there's this region that's called the vestibule. Okay, so it's like a little entryway. And in the vestibule, that's where you're going to find the utricle and the saccule. Okay, and that's where the macula are. Okay, and they're going to detect if you're moving forward and your head is still, or if you're moving your head. Okay, so what's in there is there are hair cells and supporting cells. The hair cells have stereocilia, okay, so little cilia, and then a longer cilia. And what it projects into is this membrane, and it's kind of jelly-like. So think of Jell-O, right? And it's called the otolithic membrane. Now, in that otolithic membrane, there are crystals, okay? So they're calcium carbonate, and those crystals they call otoliths. Now, there's a picture, okay? So when your head tilts, what's happening is this membrane, so this Jell-O stuff moves, and the weight of the otoliths in there helps it move. Okay, and that bends the little hairs or the stereocilia. So when the stereocilia bend, okay, it causes transduction, right? An electrical impulse is going to occur. Okay, so it increases impulse frequency in the vestibular part of the vestibulocochlear nerve, which is number 8. Okay, so the opposite thing will happen if your head is going the other direction. Okay, so let's try to show you a picture here. Okay, so here's what the macula looks like. Okay, so here are the stereocilia, so the little hairs, right? And then here's this kinocilium. Here's the gelatin that's over top of it. And then these are the calcium carbonate crystals that are over top of the gelatin. They're in the gelatin. So it kind of provides some mass to the Jell-O, so it'll move when you move. Okay, so when these move, okay, then the electrical impulse is going to be sent down this neuron, and it's going to go to this vestibular branch of the vestibulocochlear nerve, okay, which is a cranial nerve. Okay, and it does hearing and equilibrium. Okay, so those are the macula. Okay, so here's a picture of her with her head upright. So when her head is upright, okay, this is not bending. Okay, none of the stereocilia are bending. So what's happening is there's a constant signal, and it's just kind of like, I always think it was like ping, 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 ping. So it's this constant signal that is being sent, saying, okay, you're upright. Okay, now when you bend, okay, if you are bending up, you're tilting your head up, okay, the stereocilia are going to bend backwards, and you're going to get more pinging, ping, 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 ping. Okay, but then if you go the other way and you tilt your head downward, the signal slows down. Okay, so that's how our brain perceives where we are in space, is by the number of signals that you get. Okay, so that there's a standard frequency. Okay, and then when you go backwards, it gets increased, and when you go forwards, it gets decreased. Okay, and it's all about this otolithic membrane moving. Okay, now angular acceleration, okay, is at the bottom of the semicircular canals. There are these little structures that are called ampullas. Okay, and in those ampullas are these crista ampullaris, and they have hair cells, right? Okay, these are embedded in another layer of gelatin, okay, but they call it a cupula. Okay, so it's kind of shaped like that. Okay, now when the head rotates or moves, okay, the endolymph pushes against the cupula. Okay, so it makes that gelatin move. When the gelatin moves, it bends the stereocilia, it changes the voltage of the hair cells. Okay, it's gonna, if the cilia bend towards the kinocilium, the hair cell depolarizes. If it bends away, it hyperpolarizes. Okay, so it's excitation and hit inhibition again. Okay, so it's going to change the rate of firing on the same branch, same nerve. Okay, so here are the ampulla, and this is just trying to show you what they look like. So they're going to be at the base of these semicircular canals. Okay, and there's endolymph in these semicircular canals. Okay, this is the little cupula thing. Okay, so here's the cupula, it's blue, right? Here's endolymph. Okay, so that's the fluid that's in there. There's the long hair cell. There's the stereocilia, or not hair cell, hair. Okay, and then there's supporting cells in there. Okay, so now when your head spins, okay, if you're standing still, okay, it's not moving. But if you turn your head or you rotate, okay, this gelatin is going to move, and it's moving because the endolymph is moving. Okay, when that moves, it causes the hairs to bend, and that's going to trigger a neural impulse. Okay, so the information is going to come out this vestibular branch of the cranial nerve number 8. Okay, so then they're going to go either to the vestibular nuclei, which are in the medulla, okay, or they're going to go to the cerebellum. Okay, and that's going to be how we figure out, okay, if we're spinning, how do we stand up? Or the information is going to get sent to the thalamus, and then the thalamus is going to tell our cerebral cortex, so we know where we are, and like if we need to put our foot down or whatever. Okay, so this is just a picture of that, and I'm not too worried that you know all of that, but the idea is, okay, the information's coming in from the vestibule, right? And it can go to the part of the medulla oblongata. It could go to the cerebellum. It could go to the thalamus. It can go to the cerebral cortex, okay? At the same time, information's coming in from your eyes. So you know where you are. You can see what's going on. Okay, and that's that.